Thank you all for joining us today. So my name is Julia Bryant. I'm a PhD candidate at the University of Alberta, and I'm joined by my fellow co-chair and co-host, Fanor Montoya Maya, Restoration Director at the Coral Restoration Foundation. Fanor is just going to give a quick introduction to himself.
Hello, everyone. Thank you very much for joining us today. Happy to have you here and looking forward to answer a lot of your questions with this great group of panelists that we have today.
Great. Thanks, Fanor. So the goal of today's Q&A will be to share key insights and identify how restoration monitoring can support adaptive management strategies under climate change and chronic bleaching, pulling out actionable guidance that you can apply in practice. We think this discussion comes at a critical time with respect to the marine heat waves that have been sweeping across the globe. But first, we want to know a little bit more about you, our audience. Please follow the QR code that's on this slide right here to a Menti poll where we're asking you all to share a little bit more about yourselves. So your region, your role, your organization, and a little bit about your background. So we'll give you just about a minute to go ahead and do that, and then we'll take a look at where everyone's from. Okay, just noting the time. I'm going to go ahead and move over to Menti for 1 minute. All right, so it looks like we have quite a few different attendees here today. We have I'll just move to the first question. So we have quite a few people from the Caribbean, a couple people from other locations, the Eastern Tropical Pacific, Indo-Pacific, South America, a lot from North America, one from the UK, and hopefully more joining as the Q&A continues. We've also got quite a few different backgrounds in terms of positions. So we have some practitioners, researchers, volunteers, students. Awesome. And then quite a few people from the government, NGOs, research institutes and universities, and other. I'd be curious to know what the other are. You can feel free to comment that in the chat as well if you'd like. All right, thank you so much, everyone, for letting us know a little bit more about yourselves. Now we'll just move back to the introduction. All right. Okay, so I think now that we're back on this slide, we'll move on just to give you a little bit more background about who we are. So this Q&A discussion is being hosted by key members of the Coral Restoration Consortium's monitoring working group, along with myself and Fanor, who are currently the co-chairs of the group. And a significant part of this group's mission really involves improving coral bleaching preparedness and response strategies through robust data collection and analyses. But we also focus on many other essential monitoring related topics. And so if you're interested in learning more about us and what our group does, please don't hesitate to reach out and or visit the CRC's website. And one of the central products of our working group is the CRC's monitoring guide, which was published in 2020. And you can follow this link via the QR code if you're not really familiar with this document. But we'll now go ahead and break down some of the guidance provided here, which will be really key to our conversation that we're going to have through the Q&A around leveraging monitoring to limit bleaching impacts. So within the guide, we outline 4 universal metrics and 1 universal environmental metric as the most basic methods through which restoration size, composition, and status can be monitored in a standardized and useful way for practitioners, scientists, and managers alike. However, when we're preparing for things like bleaching and also responding to bleaching, we may want to get more specific with the monitoring metrics that we're using. So, more specifically, things like post impact surveys that obtain information on the spatial footprint of bleaching can really inform more targeted restoration maintenance and management in response, and may be particularly essential in deciding if, when, and how to conduct restoration post disturbance. Then, going beyond just reactive monitoring, we may want to engage with more active strategic monitoring measures that track genetic, ecological, and environmental interventions to improve reef resilience to climate change and enhance the persistence of coral reefs generally. So, specifically, these goals often aim to improve reef resilience through things like managed selection and breeding, assisted evolution via assisted gene flow and migration, and physiological stress hardening via pre-exposure and the manipulation of algal symbionts and the coral microbiome. And also, many of these methods for these approaches are still rapidly developing. And one thing that really helps with that is research. And so, the implementation of innovative methods like these, among others, to combat bleaching impacts, are really highly dependent on research-driven interventions and monitoring that focus on testing techniques for successful restoration under disturbance. And so, research-focused monitoring, while often conducted at smaller scales, is often readily analyzed by scientists to identify insights that are really key to informing restoration efforts to bolster resilience. And so these findings can then feed into a monitoring feedback loop, allowing us to better understand and apply those mechanisms that can mitigate bleaching effects on restoration. And this feedback loop is an essential component of adaptive management, which we've all probably heard of at this point, which really allows for course correction when goals are not being met. And so this structured, iterative decision-making process really requires system monitoring to understand how effective responses to bleaching and the extent, how we can craft effective responses to bleaching and also better understand the extent to which the interventions that we're implementing are actually successful. So the CRC has also more recently put out documentation that provides specific guidance on restoration under bleaching in response to the major 2023-2024 events. And the guide, which can be accessed via this QR code, outlines how to prepare, act, and monitor bleaching based on your capacity and resource availability. And so we provide guidance for when only basic monitoring can be conducted, highlighting more specifically the necessity to also monitor ahead of bleaching if possible. But while basic monitoring can be helpful, if you have the resources available, more comprehensive methods like photoquadrat or transect surveys, along with environmental monitoring, are really preferable since they lend to the extraction of useful insights from these data. And then, if possible, even more advanced methods that identify things like resilient traits, track effects on reproduction, and monitor at scale will be the most useful for discerning effective restoration strategies under heat wave conditions. So given all of this current guidance on using monitoring to improve restoration methods under bleaching, we really want to hear your questions. What are we missing here? What are the gaps? What will be useful to you? So if you could just please start adding your questions to the chat. What we'll do is we'll let our panelists introduce themselves in a second, and then we'll round up all of your questions for the panel discussion. But first, let's let our panelists introduce themselves. Today, we have joining us experts who offer really diverse perspectives on leveraging monitoring to support restoration through technological advancement, genetic tools, propagation methods, forecasting, and adaptive practice, including Dr. Lisa Karn, also joined by Victor Fox, Dr. Ileana Bombs, Dr. Derek Manzello, Jordan Pierce, and Dr. Anastasia Banashak. So first, we'll hear from Derek on how we can leverage monitoring to forecast climate impacts, specifically using NOAA's Coral Reef Watch, as well as explore the potential for forecasting to inform strategic restoration and monitoring.
Hey everyone, thank you, Julia. My name is Derek Manzello. I'm the coordinator of NOAA's Coral Reef Watch. And today I'm going to briefly introduce you to the products that Coral Reef Watch provides that you can utilize to do bleaching monitoring at your reefs. Next slide, please. So at Coral Reef Watch, we produce our own sea surface temperature data product. It's called Coral Temp. This is a daily global 5 kilometer by 5 kilometer sea surface temperature product. So you can go on our website every day between 3 and 4 p.m. Eastern time and find out what the sea surface temperature was at your reef of interest anywhere around the world as of yesterday. Next, please. So for coral bleaching, we're ultimately interested in anomalies, or how much are the temperatures compared to what they normally are. So what you're looking at here is our sea surface temperature anomaly plot. And this is showing you how sea surface temperatures compare as of May 19th, when I put this talk together, with the average or climatological conditions. Next, please. So for corals, we're interested in the sea surface temperature anomaly above what is called the maximum monthly mean sea surface temperature. So the maximum monthly mean sea surface temperature is simply the average temperature during the warmest part of the year, during the warmest month of the year. So I always like to use Florida as an example. In Florida, the warmest month in the ocean around the Florida Keys is August. So the maximum monthly mean temperature is the average temperature for August in Florida. So once sea surface temperatures go 1 degree Celsius above that maximum monthly mean temperature, theoretically, that is when corals become stressed, right? So the coral bleaching hotspot product is showing you where on the planet sea surface temperatures are currently 1 degree Celsius above that maximum monthly mean temperature. Next slide, please. Now, the hotspot product is instantaneous in that it represents 1 day of data. Now, ultimately, we want to know the cumulative accumulation of heat stress on the corals, because that is ultimately what they're responding to. Only in rare cases where you have very extreme thermal anomalies of, say, 2 to 3 to 4 degrees Celsius, are you going to see a coral potentially bleach in a matter of days, right? So for the most part, we're talking about how sea surface temperatures have been experienced by that coral over the last 12 weeks. So the degree heating week product is really the meat and potatoes algorithm of Coral Reef Watch, right? So this is summing all those hotspots over the last 12 weeks. And essentially, once you accumulate 4 degree heating weeks, that is when bleaching is expected to occur. Next, please. So we developed this product called the Bleaching Alert Area, and this basically summarizes and distills down the degree heating week product into simplified categories. And I'll get into that in a few slides. But we also have regional virtual stations, which I'll touch upon next as well. Next, please. So we produce a 4-month bleaching outlook. This is a product that's based on NOAA's climate forecast system. And this gives you an idea of bleaching risk over the next 4 months. Next, please. So NOAA's Coral Reef Watch, up until December 2023, we had 2 bleaching alert level categories. However, we ran into a problem in late fall, summer of 2023, where the Caribbean got so hot that these products basically became less useful, because as you see in this figure here, the entire basin was in alert level 2, and this was not doing a good job of illustrating just how hot it had gotten. Next, please. So we rehashed this. Again, I don't have time to really dig down into this, but again, we're now up to 5 alert level categories. I always say a bleaching alert level 5 is analogous to a category 5 hurricane or cyclone, meaning that this is when you can expect severe impacts or a near complete mortality of your reef from a bleaching alert level 5. And this is at 20-degree heating weeks or more. And unfortunately, as you can see, we've seen that level of heat stress through wide areas of the Caribbean over the last couple of summers. Next, please. So this just shows you the regional virtual stations. It's a Google Earth interface. If you go to the website and click on one of these, like Ningaloo, next, a plot like this will open up. It defaults to showing you the current bleaching alert area level there in the top left, and then the 4-month outlook in those 3 panels. So if you click on degree heating weeks, it'll show you a plot like this here on the right. And again, as you can see, unfortunately, our colleagues and collaborators in Western Australia are currently going through their worst bleaching event on record, much like the Caribbean has over the last few summers. Next, please. Single pixel virtual stations are very useful because this gives you this information for specific reef sites. So if I click on Culebra, it shows me the time series data. If I click on multi-year graph, this shows me all the data, SST degree heating weeks for the entire satellite record, and then you can compare how this current year compares to past years. Next, please. And this just shows you the link for all the single pixel virtual stations we have at US Coral Reef sites. Hawaii is in development. It's not because we don't like our Hawaiian partners. It's because Hawaii, fortunately, has been one of the few places on planet Earth that has not bleached since 2023. And I think, is that it? Do I have another one?
That's it. Awesome. Thank you so much, Derek. Yeah, that's all super useful for better understanding how we can anticipate bleaching. Next up, we have Jordan Pierce, who will be discussing how we can leverage tech and large area imagery to really better track and, again, prepare for bleaching events.
Yeah, thank you. Appreciate it. So my name is Jordan Pierce. I'm a marine data scientist at NOAA within NCOS, the National Center for Coastal and Ocean Sciences. My background's in geography and GIS and oceanography, but then also computer science. I have a big interest in mapping benthic habitats, and that's sort of fallen into shallow water coral reefs. So I'm really interested in sort of focusing software development, application development in Python. And kind of feel somewhat out of place here because I'm not really a restoration practitioner, nor am I an ecologist, but I have worked with them for almost a decade now. And instead, what I do is I focus on taking the data that is collected, which ultimately helps facilitate the analysis, those important questions that you have, and helping speeding up the process. So I often think to myself, how can I make your lives easier? That is helping you get useful information out of the data that you have as fast as possible without breaking the bank. Next slide. So I tend to focus on using things like computer vision and machine learning to help speed up the process of image annotation. For now, this is mostly just images that you might collect via scuba diving, as well as orthomosaics, which is also a form of raster which comes from structure for motion, as well as working with 3D data assets like point clouds and meshes. And ideally, we're using these algorithms to help alleviate bottlenecks, and in the context of this conversation, hopefully providing faster feedback. So that way you're able to go out, collect data, you're able to annotate it, analyze it, and then have some useful statistics that you can then report and then make decisions off of those. So there's a lot of really good software out there to help restoration practitioners such as yourself. And I think as time goes on, both those who are in the field, people who are in academia, they come up with different SOPs, and that sort of changes and sort of shifts the landscape along with changes in computer science. So things related to computer vision and AI. And these changes, require a little bit of updates and maintenance. So I think as time goes on, you should slowly adjust, and hopefully as time goes on, things become faster and faster and automated, but not too automated to the point where scientists are out of the loop. So sort of below here, we can kind of see, this is sort of a glimpse into some of the things that I work on as it pertains to image annotation, the idea of making annotation faster and hopefully somewhat more automated. So in the bottom left-hand corner, we're looking at things like coral point count or coral net. If you guys are familiar with CoralNet, CPC, randomly sampling points all over the image. This is really great. CoralNet's a fantastic resource. It has built-in machine learning to help learn from the data that you're creating. But there's also some issues associated with the amount of time that it takes to create a data set that can train a model, particularly when you're working with areas where your data set tends to be heavily imbalanced and you have rare class categories like live coral, for example, that you really need to focus on, but it's hard to do so with randomized points. So even having simple software that can basically allow you to freely place a point on these rare class categories to be able to increase your total amount of training data for those particular classes and therefore create a model that performs fairly well. And then also applying these types of models that you may have on things outside of just regular 2D images that you collect during benthic habitat surveys, such as orthomosaics, which are great because you have the whole reef in sight, and also an image. There are some artifacts that are associated with orthomosaic that make it harder for a machine learning model that's trained on images to sort of generalize, but you can see here we're applying a model on a number of patches on an orthomosaic, and it took approximately 27 seconds to do so. So there are definitely some ways out there that are sort of being developed that will sort of automate tasks and hopefully allow you to have faster feedback. The same is also true for things like polygons or segments. You can see on the right-hand side, we're in an awesome time right now where there's a number of foundational models, sort of AI, computer vision models that are coming out that make life significantly easier than it was just a couple of years ago. On the left-hand side, you're seeing the ability to one click on some sort of coral and be able to create a polygon on it. So no longer do you actually need to trace it. That's pretty fantastic, and that's a relatively new update within the past year or two. And with this sort of new technology and these ideas, the idea of chaining different models together. So maybe you have something like on the far right where you're automatically creating segments around things without any idea of what they necessarily are, but it's quite good at creating polygons around them. And then using some sort of existing model that you might have trained on patches, such as something you got from CoralNet, so that way you can classify all of those polygons to the correct species or genus. So this is sort of my bread and butter and what I'm focusing on, and hopefully we can make life easier for everybody.
Awesome. Thank you so much, Jordan. So next up, we have Anya, who will be discussing how her group has really used approaches to sexual propagation to really and leverage those in times like these with chronic bleaching.
Thank you very much, Julia. Yes, my name is Anastasia Banashak, and I'm a research professor at the National Autonomous University of Mexico. It's located in a small town called Puerto Morelos in the Mexican Caribbean. And basically, my background is in photobiology and more recently in coral reproduction, and then basically researching the techniques, optimizing techniques, as well as looking for cost-effective, basically low-cost techniques for raising corals and then outplanting them in the field, and also doing all the monitoring that goes with that. But my focus will be on monitoring coral spawning in the age of bleaching. Next slide, please. So the very first thing that you need to know before going out to monitor coral spawning is when should you go out. There are several calendars that have been published, especially around the Caribbean. These are published annually, and for example, we have a database that has almost 900 entries from different sites and coral species. And it has been used as a reference for the Greater Caribbean, but there are other calendars also published. So if your area is closer to one of the, say, the Southern Caribbean, you could use the Kamabi prediction calendar. Next, please. What we're working on, and we've just recently inaugurated the centralized database for the Greater Caribbean. So the idea is basically to include data that has been collected by a wide number of researchers and practitioners in coral spawning so that we can improve the spawning predictions, and that you can also use that database to try to work out when spawning is most likely to happen for whatever species that you're interested in your area. And that's hosted on the AGRA database website. Next, thank you. Okay, so what's happening with coral spawning and monitoring it is basically that we have to do a lot more field work. And this is because with bleaching impacting corals, we know that it does impact reproductive success. And basically, if we have a lot of corals dying, like was an example in 2023 and again in 2024, we have fewer colonies. They're generally more dispersed. And so we have to do a lot more work trying to find colonies that at least look healthy. And if you're lucky enough to find surviving colonies, it doesn't mean that they will be as productive as they might have been in previous years, because they do tend to have a reduced reproductive output. Or in a lot of cases in 2023, there was no spawning or extremely reduced spawning throughout the Caribbean. Next, please. So with climate change increasing the frequency of these events, and not just in terms of bleaching, but also we've had more diseases and more and bigger hurricanes. So basically this is impacting, the how many colonies are surviving. And so in my humble opinion, one of the problems that we're having is that natural fertilization is probably not happening for, I don't think, for any species potentially, just because of the lower coral density. Next slide, please. So basically, it does mean an increased effort to monitor spawning. So first, we are usually going out to more sites and looking to see if we can find healthier colonies. We're going to deeper sites where potentially bleaching has not affected the colonies as badly. And all of this means that there's a higher cost involved, of course. Next slide. Another approach that we have been taking, it's sort of a plan B for sexual reproduction, is to cryopreserve sperm. Basically with cryopreservation, as you know, the sperm are alive, but just in a sort of suspended state, which means they can be reactivated on thawing. We can use them at another site or at another time. Do know that bleached corals do have decreased sperm motility and egg quality. So this is another issue. If you're lucky enough to collect spawn, you may be dealing with reduced quality of larvae that are produced. Next slide. We're also now working on vitrifying larvae in my lab and with other experts around the world. It's a much more complex process, but and we have had a little bit of success. These are, oh, is that my 5 minutes up? Okay, well, never mind.
Okay.
Sorry about that, Anya. We're gonna give you more time during the questions. Okay. Julia.
Okay. Yeah. Thank you so much, Anya. Yeah. Hopefully we can get back to some of this during the question and answer period. I'll just skip through a couple of slides here, and we'll move on to Dr. Ileana Bombs. We'll be discussing how we can really leverage genetic tools for resilience.
Hello, everyone. Yes, I'm Ileana Bombs. I'm a professor and group leader of marine conservation at the Helmholtz Institute for Functional Marine Biodiversity at the University of Oldenburg in Germany. Previously, I was in the US for a long time, and I worked a lot in Florida, and I still am working in the Caribbean on coral conservation genetics. So I also chair the coral genetics working group of the Coral Restoration Consortium. And as part of this group, we have developed several guidelines and white papers to think about how we can use our knowledge of genetic diversity to help conserve and restore corals. Next slide, please. So genetic diversity is a critical component of coral resilience, and this is true for almost any species. The more genetic diversity, the more likely it is that that species will survive into the future. Genetic diversity is thus a really good predictor of species survival, and it can be used as a management target. Now, corals with their mixed reproductive mode, so both sexual and asexual reproduction, have two components of diversity that we track. It's the number of unique genotypes, or the number of clones, better is the word, the number of genets that we have to identify and track, and it is the amount of genetic diversity contained in the genomes of those genets. And long periods of inbreeding, for example, can result in a loss of genetic diversity contained in those genomes. So unfortunately, neither one of those components, so neither the genet diversity nor the genetic diversity, are visible to traditional monitoring tools, and instead we need genetic tools to analyze this kind of data. And for the Caribbean acroporids, we have developed a standardized genotyping tool and database that you can access through the QR code that's on the screen. You can go to this website, and that is a tool that's commercially available for people to just send their samples to, and then they get results back from whatever provider they use, and they can be analyzed using a standardized genotyping tool. And it'll tell them how many genets they have of Acropora palmata or silvicornis, and how much genetic diversity is in those genets. Now, for other species, such standardized genotyping tools are not yet available, but with next generation sequencing, there is a broader availability of those tools and the possibility of applying them to coral species, to really any coral species. But it is expensive, and it does require scientific computing power and expertise to interpret the data. Now, what you can do in terms of monitoring and bleaching. So if you're thinking about pre-bleaching, it can be extremely powerful to have used these genetic tools to identify how many genets are on a reef pre-bleaching. Because when, for example, the Coral Reef Watch tells you that there is going to be a major bleaching event, you can go out and collect tissue samples from those unique genets and transfer them to a coral gene bank or to an onshore nursery. And this is what was done in Florida. And as a result, we have been able to preserve a lot of the genets that we previously knew existed on the reef, and we basically saved that way. So that was a major success, but it's also a huge effort. Now, during bleaching, monitoring can help identify those genets that are in fact resilient or bleach less often than, or less severe than other genets in that population, which is important information. Now, what I think is less well appreciated is the importance to identify sexual recruits, and because without sexual recruits, the population cannot recover. And this is urgently needed information, and it will help us to then choose genets to put back out on the reef to enable sexual reproduction and make them be close together so that fertilization is possible, but also avoid putting lots of relatives close together that then may result in inbreeding and loss of genetic diversity over time. I know that's a lot of information, but I'm happy to answer or get more specific in the Q&A session.
Great. Thank you so much, Ileana, for that. Excited to get into it in the Q&A. Next up, we have Dr. Lisa Karn and Victor Fox joining us from Belize, and they'll be talking a little bit more about how they've approached conducting restoration in practice under disturbance.
Everyone. I guess my slide's coming up. Hi, everybody. Lisa Karn, Director and Executive Director, Founder, of Fragments of Hope. Been in Belize 30 years. With me is Victor Fox.
Victor Fox, Site Coordinator for Southern Belize, Fragments of Hope.
Waiting for our slides. So just a little background, we've been working in Belize on this restoration work since 2006, and on the map there, Belize is between Guatemala and Honduras, are all the sites that we work in currently. Next slide, or next animation. These are now the bleaching sites that we do, and we use a protocol developed in 1999. All of Belize uses this by Dr. Melanie McField. It's a rapid assessment where 200 corals per site are assessed for bleaching, old mortality, and recent mortality. And these maps are giving color codes for how much bleaching was observed. So we have this data going back to 2008 for many sites. This slide is 2003, where we did see some variability across sites. Next slide shows 2024, where it was just blanket bad everywhere, as most people know. So next slide. We were looking at we had a bunch of questions, how much mortality was there. So this graph is from 2023, post-2023, where we only looked at the elkhorn colonies at our oldest outplant sites, where there was under 500 colonies, and we could actually count the elkhorn colonies and do assessment of partial versus no mortality versus recent mortality. And we did okay in 2023. Those results will not be the same for 2024. Next slide. We also have been using since 2014-ish the diver-based photomosaics, which people are getting more and more familiar with now. And we currently are still using the CPCE, which was mentioned earlier. Some of our colleagues have graduated to CoralNet. We're about to learn that soon. So in this graph, you're looking at total live coral cover from 2015 before we replenished the sites. These small plots are average around 100 meters squared, give or take, and most of this coral coverage is from outplanted staghorn, cervicornis, at this site, and we do not add any corals. So we were tracking the natural growth over time. You can see we were pleasantly surprised up until post-2024, where we're back down to even before we started, because mostly it was all staghorn that grew up in these plots, and sadly now, since 2024, that is gone. So that's another way of monitoring mortality. Next slide. This again is showing from October 08. Typically October in Belize is our quote unquote peak bleaching month. That has changed over the years, obviously. 2023 and 2024 were very different. And even prior to that, we had a few years where we had peaks in November, where we had late heat stress in October as opposed to September, August, that led to peak bleaching in October. And really this is just showing what we all know, that October 2024 was the worst bleaching event that we have seen since we're documenting this. Next slide. This has several animations in it, and it's just illustrating that we had some surprising survivors, especially on the bottom two photographs. One is from Key Cocker, the one on the left, and the other one is from a nearshore site very close to the peninsula in southern Belize that is kind of murky and nutrient-rich and hot. If we keep hitting animation, we see some updated slides, updated photos. So yes, we saw bleaching, but then they recovered with this prolifera. And so we've begun spreading this prolifera around. Now this last slide on the bottom, February 25th, is a staghorn genet, and both of these, go ahead, you could do the next slide. Both of these quote-unquote winners have now been transported or transplanted to multiple different nursery sites throughout Belize. And the take-home message is, even though 2024 was really severe for a lot of our what we call inner key sites, it still seems to show and hold true that these sites that had higher temperatures historically, and we have a bunch of data to show that, are producing our few winners. And so we're moving what we're calling these winners that are more thermally adapted to multiple other sites. And that is currently our strategy. We're not effectively outplanting at this moment. We are just trying to identify, map, and propagate all of our winners and keep them going for as long as we can. And I think that's my 5, or if you want to just show our contact information, we do see some winners here and there. The last slide there, if you have questions about the protocols we use, you can contact us via Facebook, our website, or my email, the Gmail shown there. Thank you for including us.
Thanks so much, Lisa and Victor. Yeah, all great lessons learned. So next, oops, moving backwards. Yeah, so thank you all to our panelists. Now we can actually launch into the Q&A panel discussion itself. And so just a quick reminder to add your questions to the chat for our panelists. We'll be taking them in as they come in. And first, we're just going to start off with questions around what to do pre-bleaching. So how can we forecast, plan, and safeguard in this period of time before bleaching? And our goal here is really to, of course, know what's coming, line up resources, and really lock in genetic options as well as other options before temperatures spike. So now we'll launch into the questions, and I'll stop sharing these slides for a second.
Julia, we have one of our questions that was sent before, and I think this question, you may want to read it, has a culprit.
Yeah.
You're a target panelist. So
great. So our first question is, how should local managers weigh NOAA's seasonal outlook, so around 3 to 4 months, against the daily coral reef watch hotspot metric that we talked about earlier when deciding whether to trigger a bleaching response plan? So I think we now all can tell that this might be Derek's wheelhouse. So Derek, if you'd like to comment on this, that would be great.
Sure. Now, again, there is no, perfect recipe for what to look at when to determine, when to go out. It ends up being a decision based on, facts and pros and cons that need to be made. So I'll just tell you how I interpret the Coral Reef Watch products. So for the 3 to 4 month forecast, you need to remember that the skill of that forecast, or how good it is at predicting the future, gets better and better as you get closer, right? So right now, the skill of that forecast isn't great. So right now, the forecast for the next 3 to 4 months looks as good, better than it has for the last 2 years. But we need to interpret that with caution, because we are in the transition period right now between, austral southern hemisphere summer and northern hemisphere boreal summer, also called spring. So basically right now is a seasonal kind of cool period in the ocean, where the southern hemisphere is essentially off, cooling off, and the northern hemisphere is starting to heat up. So the forecast right now might be a little optimistic for what is going to happen. So I would basically keep an eye on that forecast. It updates every week on Tuesdays, and see what it's saying for your region of interest. And then when we start getting maybe 2 to 3 months out, I would start looking at the sea surface temperature plots, and particularly the anomaly plots. And the plot I really like to look at is the multi-year plots. So this shows you daily average sea surface temperature on the top relative to the climatology, which is a fancy way of saying average for that time of the year. And then it also shows you historical degree heating weeks. So this is the entire data set for the entire satellite record for a specific region, all this plopped onto one graph so that it can be interpreted. And the benefit of this is you can see how temperatures on this given day, well, as of yesterday, right? It's near real time, so it's updating as of yesterday for around the globe. So you can see yesterday, how did sea surface temperatures in the Florida Keys, in the Bahamas, in Belize compare to the historical satellite record? Are we currently tracking warmer than what has occurred in the historical satellite record? Are we average? Are we below average? And again, it's those real-time data that I'm always looking at, right? So for Florida, again, August is our usual bleaching season. I say usual because in 2023, things went crazy in July, right? So we had this very, very early bleaching event, right? So basically keeping track of those daily average temperatures using that multi-year graph, looking at hotspots. Remember, hotspots are referring to if the temperatures are stressful at that given moment. So you need to accumulate hotspots over a given duration of time before you're going to see bleaching. So again, what I do essentially is look at sea surface temperatures in near real time and consider, are they average? Are they above average? Are they hitting those stressful levels? Are we seeing hotspots starting to accumulate? Because once you start accumulating hotspots and it's looking like you're going to get close to that threshold of 4-degree heating weeks, 8-degree heating weeks, that's when you really need to start putting your actions in place. Your plans better already be in place at that point, because you're looking at a bleaching event likely happening in a matter of days to weeks, right? And again, different parts of the world may have slightly different thresholds. So for places that have seen a lot of bleaching, a 4-degree heating weeks or a bleaching alert area level 1 may not be that big of a deal for your reef anymore, because unfortunately you may have already lost a lot of the genetic diversity in the species that are most heat sensitive. So again, you really need to ideally have some background and experience and expert knowledge of your reef site in general, and be tracking those temperatures in near real time. And then when it looks like you're going to start crossing those thresholds into greater than 4-degree heating weeks, that's when you need to really start thinking about and actively getting out into the field, because that's essentially when bleaching is going to start, right?
Awesome.
Derek, thank you very much for that answer. Very comprehensive. And being as a restoration practitioner, we build the bleaching disturbance plans, and a lot of the tools that we use to assess the risk comes from the Coral Reef Watch, but it's very difficult to actually trigger. So use something as a trigger, because still there is some uncertainty, right? And it's only as we get closer to the fact actually that we know for certain if it's going to happen or not. I think there are other questions that are going to be related to that. So I think we most likely will come back to that. Julia, there is another one here that I think I'm going to put it there, and let's see what panelists want to tackle that one. And I think this one talking about gamete collection, maybe for Anya.
Yeah, definitely. So our next question is, when a bleaching alert is imminent, how do you decide which genotypes or colonies to prioritize for gamete collection and cryopreservation so that we can really maintain adaptive potential? And Anya, would you like to tackle this one first?
I have a very quick answer to that. It's whatever colonies are alive. I mean, honestly, it's been so bad, at least here in the Mexican Caribbean, that if the colonies are alive, we try to make sure we can go out and monitor it for spawning. So that's.
Oh, I think.
Yeah. Go
Ileana.
ahead, Ileana.
Yeah, I couldn't agree more. With the Caribbean, we're really in a very desperate spot. And for areas for other areas in the world where you have the luxury of much more diversity out there on the reef still, our general guideline is that you can substitute your habitat diversity for potentially adapted genets to that habitat. So if you look at your local reefs that you can get to easily, within your program, look at those habitats that all are slightly different: the forereef, the back reef, the reef crest, the lagoon, something a little deeper, something a little shallower, and strategically collect from as many of those habitat as you possibly can, with the thought that whatever genets thrive in those habitats are somewhat different from one another. And that's one way of collecting a lot of diversity.
I think we have two thoughts here. I don't know if Fenora, Anya.
No, first, Anya, please.
Yeah, just to add to that, completely agree with what Ileana says, and that's our strategy. In fact, this year we're going to more southern sites, specifically because there are healthy populations of the species that we tend to be cryo preserving. and we don't have representatives from there. And so some of them are deeper, but they're also just slightly different habitats than we have here outside of Puerto Morelos. So yeah, that's basically the strategy we do, is just try to get as many different environments, as many different genets as possible into the bank, and also to tag those colonies. I think that's another aspect that's really important when you're monitoring spawning during bleaching, is to tag the colonies, because you may get gametes from certain colonies and then the parental colonies will die. But the good news is that the recruits that we've produced are surviving, so outplanted onto the reef. So so far that's good news.
Quick follow-up there for Ileana and Anya, because we've seen some bleaching or bleach colonies spawning. Is it still worth collecting if they're bleached, even the gametes may be compromised?
I say yes. And what we do is, of course, check the sperm motility, check for egg quality, and basically decide from there. If sperm motility is extremely low, we would not do cryopreservation, but if we're really desperate and that's all we've got, then we will do assisted fertilization with those. Yeah, it's basically, it depends on just how desperate you are.
Yeah, I think the problem with collecting, low quality gametes is that you tend to get very fouled larval cultures, and they end up being a lot of work. So the desperation has to be high in order to commit to that kind of level of care for the larval cultures, for sure.
Good. Yeah. Thank you. Julia, I'm going to bring up the next question.
Great. So next, we have a very practice-driven question. So what low-cost, community-driven monitoring techniques can we adopt to detect early signs of bleaching before our annual surveys? I don't know if maybe Lisa or Victor want to comment on this.
Well, here in Belize, a low-cost method we have is engaging the fishermen and the local tour guides and giving them training to adaptive management so they can identify bleaching, and they help us a lot by bringing back that information.
Yeah, I would just add to that, of course, since Coral Reef Watch exists, we do rely on that, but we've been in this game before that existed. And so even prior to those tools, we did rely on what he said. It's a small community here. And even in the other sites we work have a well-knit fisher and tourism community. And so we get that information like real time, and we've asked for that, and we stay engaged with folks for that, because they're the ones out there all the time and report to us.
Oh, Jordan?
Yeah, something to add to that kind of goes along with the idea of having community members who are actually out in the field kind of collecting that information and providing it anecdotally. There's a project that we worked on where we're actually soliciting volunteers online to actually help us label data of coral. This is mesh voting and deep benthic coral. It's called Click Coral. It's on a project page called Zooniverse, and originally made by NASA, but it asks scientists to create projects where they basically upload their data and create workflows, and allows community members to volunteer and sort of participate in that. And we've actually found that to be very useful, both in our project as well as a couple of others at NOAA. I don't know if CoralNet has this feature, the ability to invite sort of volunteers to participate and provide labels without editing everything else, but that would be a really cool feature. Maybe Stephen can make that happen. But depending on, and this is just an idea, the timeline between when you start your monitoring, how many months or weeks do you have before that actually starts, developing a workflow where you're actually uploading a couple of images, once a day, maybe once a week, to something like Zooniverse, and actually asking people to participate and basically just provide really simple sort of census information. I will say there's a lot of people who we call them sort of armchair explorers. They really are interested in being involved in science, especially as it relates to the ocean or marine environments. They really enjoy it, and they're always looking for any sort of project they can actually get involved in, especially if they're not geographically close to coral. They still want to feel like they're helping out. So just an idea.
Thanks,
Yeah.
Jordan, for that. Julia, I think we're gonna leave one more question for the pre-bleaching topic before we move to during. And this one you may leave it to pretty much everyone, because it's what is your recommendation when reality hits, which is, funding is limited to do cryopreservation, invest in high-tech tools, genetic tools to identify, even going out to secure boats, and actually do the work of monitoring or collecting. So
Yeah, so true. Yeah. We can only do as much as the resources that are available to us. Yeah, so just in a little bit more detail, this question is, if the forecast shows a severe event but funding is limited, which one or two no regret interventions would you prioritize to protect high value genotypes? Anya?
So yes, if you're monitoring spawning, my no regret intervention would be collect from as many colonies as possible, mix them, and let them go free. And that way you've helped to overcome one of the first steps that I think is one of the most important in that has been preventing fertilization, natural fertilization in the field. So by combining from different colonies and just staying for 30 minutes, 45 minutes max, to let them fertilize, and then let that go in the water. And I think it's a pretty low cost intervention if you have the ability to go out at night. And I think it can help a lot.
Agree. Anybody else?
I would perhaps say that, if it's really low, if you really are strapped for resources, the forecast, and Derek can maybe expand on this a bit, but the forecast of severe bleaching, it is still critical that we then go out and really document where that happened. And this is important to improve the forecasting models for one. It is also really important for the management agencies to take action, knowing that it has bleached. And it is important for us afterwards, if there are any survivors, we know that they're survivors of a bleaching event, right? So that is information we can use to say, okay, there are some resilient genotypes in this area, and it was due to bleaching and not something else. Because if you leave a year in between, they might have died for whatever reason, right? So that, I think, is why it's still important, even to just with an underwater camera, take a bunch of pictures, document the bleaching actually happened.
Oh, Derek?
Can I comment? Yeah, thank you for that, Ileana. You're exactly right. So this is a really loaded topic, but the short answer is we need as many eyes and as many data points as humanly possible during bleaching events, right? And that can be as simple as qualitative observations of presence, absence, and survival. And this is really important for a lot of reasons. The big reason it's really important, again, is what Ileana alluded to, is that especially in the Caribbean, a lot of times, much of the mortality might not even be driven by the heat stress, right? So in 2005 in the Virgin Islands, a lot of the coral survived the bleaching event, but went on to succumb to white plague disease. So the other thing we see happening all the time is, corallivores, coral predators. So things like Coralliophila, which again, Ileana's done excellent work on that way back when. But the point is, you can have corals surviving bleaching and then they go on to die for other reasons. And that's really actionable intelligence to know, right? because those are thermally tolerant genotypes, and you may be able to save some of those corals by just going out and picking off those snails periodically, right? So the more information we know, the more we could potentially save. And then the other thing is, as bleaching is becoming chronic and, you know, omnipresent basically every year now, we're seeing thresholds change over time, right? And that, again, can be due to things like you're losing genotypes that are thermally sensitive, you're losing species that are thermally sensitive. So, you know, you see kind of a shift in the baseline where you might not see bleaching at a degree heating week of 4 anymore. Maybe it's at 7, 8. And we're also seeing shifts within species. You know, some species are bleaching later now. And this, you know, Terry Hughes had a really great paper about this on the Great Barrier Reef, you know, and he calls it environmental memory, right? And the question is, how long does that environmental memory last? Ross Cunning has done some work in Florida. You know, he has a paper in revision right now showing that the thresholds for pretty much all the species in Florida have increased over time, right? So there's so much more information that we can still learn, right? As opposed to just kind of throwing up our arms and saying everything's just going to die anyway. You know, what can we learn from this? You know, we really need to we need to do the work, right, to understand what's happening, because it might not be as straightforward and as obvious. And, you know, there could be a lot of hope spots that we're missing by not putting eyes on the reef. So again, the more information we have, the more we can potentially do in the future and not feel so hopeless.
Yeah, really great points about, yeah, how we can really just use monitoring to make sure that we're tracking everything, like interactions that may be occurring that we're not picking up. I don't know. I thought I saw Lisa maybe wanted to comment as well on this.
Thanks. Yeah, I was just going to add the GPS coordinates, obviously, right? You want to be able to find back your survivors, right? We said the cameras and all of this. We mentioned the community involvement and or citizen science, whatever you want to call it. The more eyes out there, the better. And I just wanted to go back to the earlier conversation, which was focused on cryopreservation and spawning, picking specific genets. Not all of us are doing that, right? Not all of the restoration groups have access to do cryopreservation or even collect spawning or do that type of work. So everything that Ileana and Ania said about choosing your different habitats and getting as much different diversity as possible for those target species, et cetera, applies to in-water nurseries as well in fragmentation, right? So if you want to stock your nurseries with as much of these, near shore, offshore, patch reef, different microclimates or microhabitats, as Ileana mentioned. So I just wanted to reiterate that it applies to fragmentation work as well and in-water nurseries. And that's the premise that we've been working with for over 10 years now. And then, yes, we coordinated when it said on the question about what's the minimum that you could do, track your survivors. So you want to be able to find them back, take those pictures and find them back. And that's all we can do right now. And I hope that's what everyone's doing. And lastly, I will just say that in terms of budgeting, we've been singing this song for over 10 years, and many people have been publishing for decades now that it's not a question of if but when, that bleaching will be annual events. And we've only seen it gotten worse in a faster manner than we all wanted it to happen. And so I would say at this point, there's no excuse not to create your budgets for bleaching monitoring every year, which we've been doing for many years. And if you're listening right now, please plan to do that, because I don't think this is going away. We don't see the political will to make this go away. And again, the elephant in the room is climate change and fossil fuel dependency. And so I don't think that's going away right now. And so what we're doing is really scrambling to try to prevent extinction or extirpation of these corals in our areas. And that's the best we can do right now in light of the fact that we're not moving off of the fossil fuels yet. Thanks.
Anya?
Yeah, I agree with what Lisa has said. I would also just like to comment, if possible, try not to fragment all colonies, but try to leave some adult colonies that are reproductive available.
We don't our rule is we never take more than 10% mother colony. And so, yeah, we're not going to be moving. Well, I would say there might be some exceptions at this point with some really scarce, rare species that you might want to move the entire colony, but that's a whole nother discussion.
Yeah. And maybe not all groups have the same criteria that you do. So just maybe either try to leave a few adult colonies undisturbed or take minimum fragments from them, because especially in disease and bleaching environments, a colony that has been fragmented, may have some consequences in terms of reproduction.
Thank you, Anya, and thank you, everyone. Julia, it's okay. We move to the next section, and I would like to say that we have a comment that I think is spark a good question for the during bleaching event from Joe Hillman. He said that from very limited funding, Dominica, with no ability to crop preserve, etc., we install shades over as many high value genotypes as we can. So this is a very reactive, and this is one of the actions that is triggered from time to time. So, and we have a question here that if anyone, and this is more for the practitioners, if you have monitor and have evidence, I'm going to bring the question here. So I'm going to ask Julia if that is okay. What evidence do we have that proactive shading or localized cooling, some examples there, meaningfully reduces bleaching severity at restoration sites? Have you monitored that? Is there evidence on a restoration context, I'm saying, not on a lab context or setting? Lisa?
Hi, thanks for asking. We have not actively shaded anything or done any implementation such as this, nor do we plan to, for multiple reasons. A lot of our sites are offshore, rough weather, logistics, etc. However, we have noted, and I don't know if there's time to discuss it further, that many of our winners or survivors are located in turbid areas. So they have this sort of natural shading. And although, ironically, they're also our hottest areas, as my slides were like, they were almost 34 Celsius compared to other sites. They also had that sort of natural shading from the turbidity. And it's not just in Belize. We've seen from Tela in Honduras, a nearshore beautiful stand of Pomada that's still living. And of course, years ago, I forget the name of the Colombian reef that was near the port, that was near shore. It was a big surprise with all this wonderful coral cover, and that was super turbid as well, right? So it's just something to think about, that there is definitely, we already know that light as well as heat is a factor for bleaching. And so whilst we don't particularly do shading, I'm just illustrating the natural environments where there is that sort of shading from the direct light. So we've seen that work, surprisingly.
Thank you very much for that. Julia, with your permission, I'm going to bring up one question that challenges technology. So
right.
During bleaching.
Yeah. So now now to the panelists, and maybe one also in particular. How can we, how rapidly can structure from motion, so SFM surveys, be turned around, so from field capture to live coral cover metrics, so managers can confirm bleaching severity within days rather than months? And I don't know, Jordan, if you'd like to comment on this.
Yeah, for sure. Well, so for structure for motion, sort of the more classical sense, if you're using something like Metashape or Pix4D, those particular algorithms are going to take a bit of time, right? The data collection itself takes time. The data processing, actually running through the workflow, takes time. The more images that you have, the larger area that you're trying to create or reconstruct requires larger computational resources. These things take time. But luckily, actually within the past 2 years, and also even a little bit more recently, there is a lot of research that's sort of re-emerging more interest in applying deep learning techniques to 3D scene reconstruction. This is not something that's currently in Metashape or Pix4D. It's still an active area of research. It's being published. There's demos online that you can find on places like Hugging Face or Hugging Spaces. And although it's not technically commercial software or not really at a point right now where it can be used in the same way as a drop-in replacement like Metashape, which is what people use, it does sort of demonstrate that we're currently on the path to having faster ways of getting the same data products that you normally would from a typical structure from motion workflow. So for some of those of you who are really interested, you can Google this at your leisure. There are techniques called Duster, followed by Master and Faster. They're all spelled sort of funny with a 3 instead of an E. These really are taking some pretty state-of-the-art techniques using large vision foundational models to be able to, in very quickly, we're talking like a number of seconds on a standard GPU, create a 3D point cloud with up to 100 images. Some are able to create larger scenes using larger images. Some are able to do slightly higher resolution 3D models, but take a little bit more time. But the nice thing to know is that we are in a direction where hopefully in the near future, there will be available commercial products for people such as yourselves who are practitioners that you can use as a drop-in replacements for structure from motion. So that's one thing that's actually like, creating the structure from motion data. But there's also a couple of researchers who have sort of built off of the current workflow that most people use. So 2 people in particular, and this doesn't necessarily speed up the process, but you might be able to get your results a little bit faster, which is great. One is, his name is Jonathan Sonder. He actually gave a presentation not too long ago, and his paper is scalable semantic 3D mapping of coral reefs with deep learning. He actually developed a GitHub repository. So if you code, you can look up deep reef map. And he basically shows how using images from a GoPro that's collected, being able to create a 3D point cloud sort of on the fly in real time and actually applying what's called a semantic segmentation model. You can think of this as for every single image, every single pixel in that image is provided with some class category. And then actually projecting that onto this point cloud that's being generated. So in a very short period of time, you can imagine just sort of scanning over the reef and actually having a 3D point cloud that's classified at the real time. And these are still sort of research projects. There's code available that you can sort of play around with and try to adapt it yourself. But it is definitely sort of a nice thing to see. Just shows that there are researchers who are trying to make these things go faster. Another one that's really kind of notable is Rapid Benthos. This is made by, I'm going to butcher the name, it's either Teeny or Tiny Remmers. The paper itself is titled Automated Segmentation with Multi-View Classification of Coral Reef Communities from Photogrammetric Reconstruction. And the idea is you already have your structure from motion workflow completed. You are now working with an orthomosaic, which is what a lot of people are moving towards, right? It used to be benthic, habitat, quadrat surveys along the transect. You're collecting images, you're doing coral point count, maybe coral net. But a lot of people tend to be moving towards structure from motion, where you go back to the same plot on a routine basis. You map out that entire plot using structure from motion, and you create your data products. What do you do with those data products? Well, with an orthomosaic, you have the entire site all within one giant roster. Now you need to annotate it. That takes a lot of time. There's a lot of tools such as TagLab, which is great for doing things like that, creating polygons and also points. But this particular paper actually looked at using other foundational models to automatically segment everything in the orthomosaic. These segmentation models, maybe you've heard of them. They're called SAM, Segment Anything Model, and other variations, where they're trained on just a lot of imagery data. And whenever you apply them, they actually just create polygons around things. So you can think like whole coral, not necessarily sand or macroalgae, but things that have a definitive boundary where they start and where they end. It's very clear that this is one object. And that kind of alludes back to the slide that I'd shown previously, where you can simply provide a point and automatically segment that. So there are techniques out there that are being developed that are being applied on top of existing workflows that a lot of practitioners might be using. to help sort of speed up this process. In terms of real time, actually collecting the data and actually having like an onboard computer processing that data, so whenever you pull out, whatever device you're using, actually having results, I think that's still a bit ways away. So, yeah.
Thank you, Jordan. We're gonna have two more questions here, Julia, if that is okay with you. I'm gonna bring one that touches on what Jordan mentioned, and pretty much what everyone was talking. And I think, as you know, in Florida, well, in Florida and the Caribbean, we consider taking corals out of the water, to keep to safeguard some genotypes. What Lisa said, identifying those areas of high turbidity where we find some pockets of or correspond to drivers of resilience in the Western Indian Ocean or Fiji and other locations. They just look for areas of some cooler water as well. So I think this question is, how can we use real-time monitoring to identify those pockets or drivers of resilience so that the practitioner, can focus the work during the bleaching event? Any comments there, answers? Derek?
So one of the things at Coral Reef Watch there are the single pixel virtual stations I mentioned. So Eric Geiger, who works with Coral Reef Watch, these were kind of always in our plans, our 3-year plan. But he had the brilliant idea early in July 2023 to stand these up immediately for Florida. And we've received a lot of very positive feedback about these stations as we've developed them for U.S. coral reefs, essentially for key reef areas and nursery locations. And the benefit of these are that you can essentially cross-compare, different nurseries, different reefs in your region directly, as opposed to trying to, dive in and tease out individual data points from the Coral Reef Watch data product. So the idea being, you can triage your responses essentially. If you need to do coral rescue, then which reefs are, essentially the hottest, the earliest in time, which reefs may not be hot, and you can kind of, come up with a tiered plan for your responses to know where certain things are worse than others. Also, potentially you could look at the history of the data and say, oh, with this reef, for whatever reason, doesn't look like it gets as much heat exposure as this other reef. So again, it's kind of a tool that can be used to understand relative amounts of heat exposure in near real time, so that, we don't have a repeat of what happened in Florida in 2023 when, things got really bad really fast. And there was a general panic to do everything, everywhere all at once, right? So the idea is hopefully that in the future, we can learn from that event and understand, as these marine heat waves develop, where to prioritize resources. Because, hypothetically, you could have a bleaching event happening off Key West and, off Key Largo, it could be totally fine. Again, that's a hypothetical, but that theoretically could happen.
Thank you very much. And I'm going to bring up the last question, Julia.
Awesome. So yeah, given all of these amazing tools that we can use, given your expertise and experience, how would you adjust your monitoring approach during the next bleaching event?
I think this one is for Lisa and Victor.
Yeah.
Sure, no problem. We've been adjusting since 2023. So as I mentioned previously, with some exceptions, our peak bleaching in Belize was October usually. In June for us, it started, in 2023, it started for us in June, not July, as you mentioned in Florida. So we have been doing nonstop bleaching surveys since June 2023 monthly, whereas prior to that year, we kind of did September, October, November, kind of looking for before, during, and after. So what we've already adjusted is our annual training approach, and we've actually doubled up on that. We can only do maybe 11 people at a time because it's hands-on. We do a 5-day protocol, and no longer are we teaching the replenishment methods that we used to teach yearly. We're now focusing on oral ID, and as I mentioned, the rapid swim bar method that we use in Belize to identify 200 corals per sites. They need to know their species. They need to learn how to identify pale versus partially bleached versus wholly bleached versus diseases versus it's pretty in depth for a lot of people because there's so many nuances. Derek mentioned predation, so it's a lot for people to learn how to distinguish what's going on with a single coral head, and then also your percent old and new mortality. So we've done what, 30 odd people. We've done this 3 times since 2023,
almost 40. Yeah.
Almost 40 now, and we did it a few years back too in 2008 and 2018. But that's our focus now, is to get, as we talked about, more eyes and qualified eyes out in the water, and adjusting it for us just means getting more funding to do more teams out there, which we're slowly doing. Instead of just our single Southern Belize-based team, we're able to help fund other groups in our National Coral Reef Network that we have in Belize, which includes fisheries and the University of Belize and other NGOs. So getting multiple teams out at the same time has been our focus because we have a lot of reef to cover. So that's how we've been adjusting since these severe events, is by getting more qualified people to know how to do our standardized protocol. Again, I can share that link somewhere. That is really a rapid way to assess the severity and the extent of the bleaching at the same time in multiple locations in Belize. You have anything you want to add?
No, you covered everything.
I'm sorry.
I maybe would like to add something that hasn't really been so much in the front of my mind, but has recently really become central to how I think about coral restoration and conservation in the Caribbean specifically, because we're so far down the road. But this, the only way these populations are going to survive and potentially adapt is if they actually successfully reproduce sexually. And we don't have a lot of good monitoring data on sexual recruits for the acroporids. Part of that is because they recruit very rarely, and part of it is because it's hard to identify small recruits. But don't give up on surveys for recruits, because if we do find they're recruiting somewhere, that would be a really high priority area for conservation measures to make sure that those recruits survive and grow up into adult corals. And it would really tell us a lot about where might they be coming from, and how can we move forward, because that is not working. That's the sexual reproductive cycle is broken, and we don't really truly know where it broke and what of the many points that it could have possibly be failing. Where does it really fail? And so we definitely need more information about that, and monitoring is a large part of how to get that information.
Definitely. Thank you very much, Ileana and Lisa, for answering there. I would like to bring some comments that we have here before we move to the next session. As we wrap up, we only have 5 minutes left. Andrew Ross suggests looking at submarine springs, that we may find, some survivors there. There's coral reef there. I think that's an important future coral there, maybe. And Austin, thank you for joining, Andrew and Austin. A bit of every remaining genotype of all red coral species can go into a tight gene bank nursery that is easily shaded, and duplicate nurseries can be created as insurance. I think that those are very common. Thank you very much for sharing those. Julia, happy to move to you. I think on the last 5 minutes that we have on the post-bleaching recover, evaluate, and adapt, right?
Yeah. So,
and I think I'm going to bring, because matter of time, this will leave us, I think, to the closing. I think
yeah.
I think this is a very relevant question to all of us, because there is a lot of work happening now, right? And after the bleaching event in Florida and right now in other parts of the world, a lot of that sparked a lot of work, publication, and everything. But sometimes it doesn't come as fast as needed for decision making or to apply those findings. And I know it's not only the researcher's fault; it's the publication processing that takes reviewing and all of that. But could you provide some guidance on these questions for the audience?
Yeah, Ileana?
So I think this is exactly what the CRC was intended to do, right? Is to be able to make the communication between all these different people, practitioners, divers, restoration folks, managers, researchers, a little less cumbersome. And these webinars are one way, right, where we can organize them on a much shorter time frame than it takes to publish a data-heavy paper. And the CRC website also contains a number of white papers that have been put out. The working groups are available for contact if there are questions or if there's data that can be fed back to the working groups and be distributed that way. Now, these are all not perfect. I wish it was more efficient and we would have a broader, a more easy way to reach everyone. But, that is really what the CRC was intended to do. And if there's folks in the audience who have ideas how we can make that more efficient, I think we'd all be super happy to hear about it and implement it. And I know that there's some tech folks out there who could help us with getting the communication tools up and running on the CRC website as well.
Yeah, really great points. Anya?
Yeah, I think also make use of social media. Just make sure to report in a non-technical sense for people, and also to make sure that you're reporting as precisely and scientifically as possible, but keeping it to a level of language that anybody could understand. I think also Coral List is another source or another area where people tend to publish their findings when it's something a bit more urgent. So I think there are different ways, and through CRC we can do a fair bit of social media as well. So I think those are ways to get information out very quickly.
Definitely. I think that's what the CRC is here for, and I think this, as Ileana said, this is the purpose of the community of practice, right? Being able to share faster, than the traditional avenues, and maybe even in a language that is more accessible to everyone as well. With that, Julia, I'm going to bring up this question, I think, to wrap up, because we have so many tools, so many information that is being collected. It can be, definitely we need to collect more, right? But I think this one is, how can we actually use all that information to assess future risk and better understand when, where, and how to act? And I think this is to wrap up, everything that has been discussed: tools, protocols, literature, and resources, and new avenues of research and everything. But how can we collect that? The CRC is one opportunity, but any more around putting everything together into something to act on?
Lisa?
Hi, I guess I'll try to answer this briefly. One of the mantras I've been saying over the years is if you're engaging in any type of reef restoration activity, you need to know your reefs. So, for example, I lived in Belize over 10 years before I began this project. I didn't just like show up and do my thesis here out of the blue or anything like that. And I spent a lot of time underwater in the tourism and research aspects before we even began to think about restoration. So I would say this applies to everyone in their sites. You need to know your sites, and then with the caveat, unfortunately, even in our case where we have lots of long-term monitoring data, the last couple of years have really literally been on the charts. And it seems like from everything I'm reading, there is no more to normal or patterns or anything like that. So it's kind of a tough question to pose out there, because you have to sort of know your history of your reefs and know what's been going on. But then with a small caveat, like I said, even with some of our more resilient sites, we saw massive problems with the last two events in 2023 and 2024. But again, that is also those hotter areas where we found our few winners or survivors. So again, Belize is different. We're the second longest barrier reef system in the world, and so we're nothing like Florida. We're nothing like Jamaica or fringing reefs there. So we have a lot of different environments. So I'm not sure my answer is really applicable to everyone that's watching or listening. But again, the two things I think is that you can't just jump in out of school and know something in a couple of years by looking at some models. You have to really engage people that know your reefs, that have long-term underwater presence in the reefs. And then again, sorry with the caveat that climate change effects are happening so much more rapidly and severe than even most of us thought. Although we expected it, we didn't expect it just like this right now. So it's a very tough question to ask, I think, or to answer rather, to answer. Do you have anything you want to add?
Yeah, thanks, Lisa. All really great points. And yeah, of course, everything needs to be taken in a case-by-case basis, and really need to consider like the environment in which you're kind of coming in and trying to conduct this restoration. I think we're running a little bit out of time, so I think it's time to wrap up. I'll go ahead and just share a final few slides because we'd really like to hear what everyone took away from this discussion. And so I'll just pop up the last few slides from our presentation here. Yeah, so now that we've discussed what to do during bleaching and following bleaching in terms of capturing lessons, rebuilding stock, refining strategies for the next heat wave, as well as what we can do ahead of time and all the tools and insights that our panelists have provided some thoughts on. We just want to hear what everyone's key takeaways were from this conversation. And thank you again to all of our panelists for their valuable insights, and again to you for your active participation. So if you could just follow this QR code over the next minute, we'll take a look at what everyone thought or what everyone pulled out of this discussion. And then before we pop up the results from your answer to this question, we just want to make sure that everyone checks out the Coral Restoration Consortium and feels free to get involved with the CRC as well as our working group if you're interested. You can follow this QR code to our page on the CRC's website. And we also really want to encourage everyone to suggest more topics that you'd like us to cover in future Q&A sessions or targeted conversations, as well as please do feel free to reach out via the CRC website or our social media channels. And yeah, thank you all so much for being part of this really timely discussion. And we look forward to seeing you all at our next event. And if you all want to stay on, I'll just look at pull up the Menti results around your key takeaways now.